Yes, and to answer this question right from the start, I think there's a pretty good chance that we can, based on work that's been done over the past decades, but also during the current round of research that we've been hearing about, some really exciting results. So we're now experiencing our fourth outbreak, as we've heard, you know, there's been successive programs starting in the 1970s to investigate this problem, and you know, you could be forgiven for asking yourself the question, why haven't we figured out how to stop it? Well, and I kind of think that we have, but we just need to focus all this knowledge that we've gained into a plan and be ready to act on it when it's necessary the next time around. So, what have we learned so far? Well, quite a lot. Still haven't been able to stop these outbreaks from starting. It's a tough problem, though. There's multiple causes to these outbreaks. Most likely it's a combination of nutrients, which are probably always above the threshold level to ensure larval developmental success. It could be related to a reduction in the number of predators, but it's probably also fundamentally related to the fact that crown of thorns is the most fecund starfish on the planet. So it produces more eggs than any other starfish. Guinness World Record holder, producing at least 100 million eggs per season. That's recorded so far, and for bigger starfish it could be twice that much. So you know, once you start to let these things get away, there's no way to stop them. But we've also been getting a deeper understanding of these outbreaks. There were indications back in the 90s when I was a postdoc working at Ames that there was a cyclic sort of predator-prey thing going on with crown of thorns. That's what modeling that Russ Babcock and Roger Bradbury were doing indicated. And even back then, there was indication of the COTS initiation box up north of Cairns. And subsequently, you know, those predictions have been shown to be correct. You know, the outbreaks have always started there in that area. They've started around that same cycle of timing, and we know we can detect them. So I remember hearing from my mates who were in the LTMP in the mid-90s saying, we think there's going to be another wave of outbreaks because we're starting to see clusters of starfish in this initiation area. And we know we can detect them even with divers at pre-outbreak densities around here. So this is an example from just before the current outbreak. And with the kind of technologies that Sven's talking about, you know, we've got even more tools to detect these outbreaks just around the time that they're about to start. So if we do detect them, what can we do? Well, we've been developing a much more effective control program than has ever been implemented before as a result of a whole range of work, from the development of the single-shot technology to the whole integrated pest management approach that's using dynamic thresholds to help people focus their effort more effectively on when to, how far down to take the populations, to what density, and when to stop. And that's now going to be applied in a bigger fleet of boats to systematically protect coral around the whole reef. So, for example, we know that at lower coral cover, you need to have a lower threshold of crown of thorns. So at low coral cover reefs, this fleet's going to be able to work harder to achieve coral protection. Whereas at higher coral cover reefs, a higher threshold density of crown of thorns is required to get to that break-even point where the crown of thorns are no longer driving coral density down or coral cover down. So all of that's starting to take effect now, and we're really getting better at it. And we should be able to apply these improvements in our ability to control starfish populations to stopping the next outbreak. But really, the problem has been in the past that there's a cycle going on outside of the reef as well as inside of it that's impaired our ability to do this. And it's been noted previously, Brian Lassick wrote this paper back in the 90s about being prepared for the next outbreak. And even earlier than this, there were reports that were written by people in the 70s that were saying pretty much the same thing. They said that we had to do something, but they really weren't sure what that was. So this cycle that's responsible for the kind of lack of action at critical times at the initiation of outbreaks is pretty much like this, where the coral comes up, then the crown of thorns populations start to take off when there's enough food, but the funding to actually do something about it lags even further behind, so that by the time we're at our peak of funding, there's really very low coral cover, few crown of thorns, and it's certainly too late to do anything about that particular outbreak. So we have to get these things to align a lot better and potentially have a funding line that's constant through here, as Dave was saying earlier today. And another kind of improvement of our knowledge about crown of thorns populations that's going to help us do that effectively is an understanding of how density-dependent effects in crown of thorns reproduction can be used to focus that control effort at the pre-outbreak stages. So if we double down on our monitoring, we'll know where to look and detect these early outbreak populations. But when is it too many crown of thorns at that low density? What's the threshold level for stopping an outbreak? Not just threshold level for crown of thorns out eating the growth of the corals. Well, some modeling that's been done, and Jacob Rogers will talk in more detail about this later in the conference, shows that we need to keep crown of thorns below about 3 per hectare to limit their reproductive success, and we need to stop them from forming aggregations. So there are 2 plots here with successful fertilization on this axis, and you can see that this is a random distribution of starfish, and these are successively more aggregated starfish populations where they're clumping together. And you get this real acceleration in successful fertilization when you get high levels of aggregation, even at quite low densities. By the time you're getting up to traditional outbreak populations, the improvements in fertilization are not really increasing that much more. So this is where you really need to get to them to stop them from being successful. And you can also see, when you look at the total number of larvae produced in a population, that at this kind of medium aggregation level, you get quite a pronounced acceleration in reproductive success once you get over fairly low densities here. So that red line is starting to diverge from the other lines at about 3 per hectare. So down here, we're really getting a limitation in reproductive success, particularly if you can stop them from aggregating. So there's a reasonable chance, I think, that if we get in there and get to those populations in the initiation box, stop them from getting into these parts of the curve, we can stop them from producing a lot of larvae and propagating that next wave of outbreaks. We can probably do that with the diving teams that we've got now, because we are getting really good at it and we've got efficient means of doing it. But we may not need to do this forever. Probably we'll have to do it for the next outbreak. So in the long term, we need to understand more about these non-outbreak populations, to understand their ecology, how they interact with predators, and how they interact with each other, the starfish. Perhaps using things like, green zones to keep the densities down, because predators don't have to eat very many starfish at these low densities to make a difference between 4 and 2 starfish per hectare. So to get them below the threshold. 2 starfish per hectare is not going to stop an outbreak that's already active, but but it can stop an outbreak from getting to that threshold. And we can also use other technologies to disrupt those populations, like push-pull technologies that could break down the aggregations, or also gene technologies that could produce sterile individuals that would reduce the reproductive success. Something that people are probably familiar with here in Cairns, where similar technologies are being used to control disease vectors like mosquitoes. So, in summary, I think the outbreaks are probably going to continue because of the very biology of these animals. They're so fecund and they're just built to have outbreaks, but we probably can reduce the frequency of them and perhaps prevent them from occurring for quite long periods of time. I think the crown of thorns obviously is essential in a wider reef restoration program to protect those valuable corals. We've got technologies that can control pre-outbreak populations in the short term, but a lot of other technologies show promise, as does the implementation of green zones. And we need more effort put into that non-outbreak phase of populations to ensure that we don't let the next one get away. Thank you.